medical diagnostics startup. We're based in Mountain View, California. We're about 2.5 years old. Our first product is called the Scout. It just went on pre-sale on a website called Indiegogo, which is a crowdfunding website. It's raised about $850,000 in pre-sales in the past 3 weeks. What we do largely is take a top-down approach to inventing new medical diagnostics products. So instead of being sort of a solution source of a problem, so a lot of times you'll see an academic lab will come up with a technology for medical diagnostics, and then they'll try to commercialize that technology. What we do is look for problems in the medical space, and then we try to find a solution or invent a solution to those problems. So we'll identify a problem, do a literature search, and then try to develop a product that solves the problem. And so what we've challenged this group with doing is to come up with a way to make a really compact, consumer-friendly device that can copy DNA. So there are several different ways of copying DNA. One's called polymerase chain reaction, another one's called isothermal amplification. And in isothermal amplification, you heat a droplet of liquid that contains a bunch of enzymes in the DNA you want to copy and DNA building blocks, and you essentially magnify or make more copies of that DNA so that it can be analyzed. You can use this to check for infections, bacterial infections, viral infections. So these guys prototyped a very nice heating system that you can use to heat small volumes of liquid. And let me take it from here.
We're Group H, and we were tasked with creating a blood test or a USB stick form factor this quarter. So our objective was to design a circuit that could diagnose the presence of bacterial, viral, and parasitic DNA sequences within a patient's blood sample. Now, obviously, there's devices like this on the market, so the further requirements that helped us separate our device is that it needed to be controlled and powered through USB, needed to be small, and also we wanted it to be user-friendly so a non-medical professional could use it in their home. So, as we mentioned, there are other devices out there. Many of them rely on complex biochemical processes that in turn require costly components. Also, many of them are just created for lab environments. For example, this is the Lupin by the Mass Group. As you can see, it's probably not something that you'd want to take with you to a remote location to do point-of-care testing. So the advantages of our device is that it's going to be inexpensive. We're going to use a simple process that can be completed with very simple circuit components. In addition, it's going to be fast and portable, which makes it an ideal point-of-care testing device. And on top of that, we also created a software program that the operator should find user-friendly and intuitive to help them perform the test. So a little bit of the background, Dr. Rowe had mentioned it previously. We're actually using the LAMP technique, which is the loop-mediated isothermal amplification. So kind of the steps that are involved in this is you mix the DNA sample with primers. These primers are specifically targeting a certain DNA sequence, such as the viral infection that you would want to diagnose for. What you then do is heat it for approximately 30 minutes at about a constant temperature between 60 to 65 degrees Celsius. Now, if the targeted DNA sequence is present, replication will occur. Finally, at the end, you'll be able to add a detection agent such as cyber green dye, and based on the color that's produced, you know whether you had a positive, a green, or a negative result. So here's kind of our simple system layout, just to give you an idea of what's going on. We're using the Arduino Nano just because it's very small. That's kind of acting as the brains of our circuit. It's communicating with the PC. Also, it's controlling our sensors and reading the data from our light and temperature sensors. And it's also controlling our heaters. You'll notice there's 2 heaters. The reason for this is heater number 1 is the more powerful of the 2 heaters. That's used to get the sample up to temperature quickly, and then heater 2 is in kind of a cycle phase where it then maintains the temperature throughout the process.
So after final night system development, And in addition, we have to learn all these new computer software to finish this device. We learn the processing Arduino for the programming, and we also learn EcoCAT to design our PCB. And we use the SCANADU and the Solomon to finally design our solution.
Well, among those challenges, the biggest challenge in our hardware design will be the selection of the leaders. And the reason is that because there's a lot of limitations for the mobile device, there are power device, there's some current limitation and limited space. We do lots of research on different features, and we finally decide to use a power resistor as our features. The reason is that at first it's pretty small. You see, compared to a coin is still very small, and it also has very high energy transformation ratio. Also, it's pretty cheap, so we finally decided to use it as our features. Here is our circuit design. Basically, we can separate our circuit into 4 modules. A microcontroller, which is used for controlling the whole system. The temperature sensor, which is used for detecting the temperature. The current sensor, which is used for detecting the current. In addition, we also have a heaters with an electronic switch. The reason of us to have an electronic switch is that the pin from the Arduino cannot offer high enough current. So we decide to power our heater directly from the power pin and use electronic switch to control the on and off. Here is our EcoCAT board design. As you can see, it looks a little bit larger than the ideal one. And the reason is that we put extra holes on it for testing different configurations. And we also label different regions on the PCB board such that it will help us to do the troubleshooting.
So here's actually PCB with a lot of components on it. Thanks for Chris' outstanding soldering skill. We have all components soldered by design. So as you can see, there's a color sensor standing on the top of the heaters, and the infrared temperature sensor is facing at the heaters from the side. So after the very beginning of this course, our mentor, Dr. Rowe, told us we can use the 3D printer to make the exposure of this device. This really gives us a chance to finish our project from scratch to manufacture. So here is our sketch using Google SketchUp. We designed this as a rectangular box because a rectangular is easy to carry and easy to store. Also, we can set
first open up the program, it'll give you a splash screen, just so you know what's going on here. And just click through, it'll welcome screen here, and it'll instruct you to click the logo to go on. From here, you want to insert the test sample, and as you see, we're inserting the test sample to be heated. And as you put the test sample, it tells you to click along, and click start to begin the process. It takes 30 minutes to set down. You can see here one turning on right now. It's time to go down. We're going to speed up this process. You can see that it gets up to temperature rather quickly, definitely under 1 day. And now heater 2 is kicked on. And this heater 2 is going to be used to try to keep the temperature at constant 63 degrees Celsius. And here will be the end time of the 30 minutes. And now it prompts the user to put in a dye. When you put in the dye is actually a clear substance. If it actually positively reacts, it's going to be green. So we put in a green sample there just for the color identification to know that the test sample is positive. So you see here, green would be a positive result using that specific dye because there are two options of tests we want to be using. And now it's processing the color. And as you can see, it says that the test results are positive. So in summary, we designed device capable of performing a lab process. Free software that interface this, make it very user-friendly for the user, and able to detect 2 different types of assays, all while meeting requirement size strips.
So the color sensor is actually, it's a group of photodiodes that have filters over them. So you have RGB filters on them. So you don't actually get the color directly from it. What we had to do is it converts it to a frequency that is in no way related to the color. We then use that frequency in order to kind of come up with a classifier. And then so when the test sample is put in, we then get the values from the test sample and compare it to our classifiers in order to determine if the color is positive or
negative. I have 2 quick questions. One is that the printed circuit board you showed, did it work?
yes, it did work.
The last time